Hello and welcome, dear friends. Let's continue our quest to understand the DNA repair with respect to the methyl mismatch repair. MMR, it stands for methyl mismatch repair. In this presentation, I have taken a different approach. First, I would like to just give you in brief, in a somewhat light humorous form, what this whole thing which you are seeing here is all about. This is the circus which all these enzymes assemble and enact to do the repair system which is for the damage done on the DNA. So first I will just like to show you in a humorous way, in a cartoon way, how this thing happens. Then we will go to the animated version. I will explain it with help of animation. The third part will be, I will not talk, only with a musical background you will see the animations. And lastly, with PowerPoint slides, I will explain what is mismatch repair or the methyl mismatch repair. So first, in a light humorous way, let's see what this is all about. So if you see here, this whole thing, there is a mismatch, means adenine and thymine, these are the normal base pairings. There is a methylated thymine, which is methylated adenines are there. There is a thymine which is undergoing a base change. Thymine gets converted into uracil. So this is it. Okay, now see here, just take it very lightly. No science here in this slide. Just understand it in the form of a cartoon. Okay, so you see here, this is a damaged DNA. First of all, a protein called as mut-S. Mut stands for mutant, mutator gene, mutator where there is a mutation or something to do with mutation. So mut-S, it just says, hey, I have found a damaged DNA. So this second scene you see, so it has an affinity for a damaged DNA or a mismatched DNA. How this thing happens, when this thing happens, how it sustains, you know very well. I will be explaining it in the future slides, but here you just see, so it goes and sits there on the mismatch. So it is having some affinity. It is a protein enzyme sort of affinity towards a damaged DNA. Second, it invites mut L and mut H. So this is a mut LH protein. Hey, come here, it says. So it has a affinity, mut L and H has a affinity, or it goes only by invitation by mut S, which has already gone to the spot of damage. So it is just like a team. The team first investigates, officer goes, then he calls up to the project manager, then a coordinator comes, then the principal investigator comes, and then the main what you can say mechanic comes. Then he comes with his tools and box and he cuts and pastes and damage is done, and then everything is okay. So just like that, first it goes and sits, mut S invites mut LH, come here, it says, then mut LH and mut S, they sit there at the spot. Then mut LH is scanning which is to be cut. It decides which is to be cut, and it says, hey, this is the wrong base. Then okay, I will cut this, and this is UVRD product, which is a helicase plus a exonuclease. Exonuclease is one which is capable of cutting single stranded DNA. So all these assembly proteins, these proteins, this assemble at that spot, cut, repair and ligate and finish. This is over. So this looks so simple, but as you can see here, this is the whole circus which is going on. It is not as simple. There is a bend, there is a loop formation, and it's very interesting. The whole thing is very interesting. So I will first show you in an animated version, then without talking I will show you some animations, two animations of the same process I will show, and then finally we will come to the PowerPoint presentation and in detail at the molecular level how this thing functions. So I hope you enjoy this repair of damaged DNA by methyl mismatch repair. MMR stands for methyl mismatch repair, and the DNA methylation system during replication, if you recall, that has a very important role to play in this. So come, have fun, let's see. So come, let's take a look at methyl mismatch repair system. But before actually going to the repair system which takes place in eukaryotes and prokaryotes as well, here example is in E. coli, I would like to remind you of a system which already exists in the prokaryotic system, that is called as the proofreading or the editing function of DNA polymerase. Now this DNA polymerase, when it is moving along the fork during replication, see here, these two black lines are the parental DNA molecule and this violet line is the newly synthesized DNA. Now what has happened here is a mismatch is remaining. It has not been corrected by the DNA polymerase system. Why this has happened is because one base in every 1 million billion bases, that is 10⁶ bases, there is a chance that DNA polymerase will not correct it or it will overlook it. It will just leave it as it is. So this is the case where the methyl mismatch repair comes into action. Those mismatches which are overlooked by DNA polymerase editing function. Now, how does DNA polymerase recognize it as a wrongly incorporated base? You know, methylation. Methylation is the main step by which DNA polymerase recognizes it as incorrect. Now, the same DAM or the deoxyadenosine methylation system is used here for the mismatch. We will see that. So, see here, this black, if you see two yellow dots here, these both strands are methylated. Here what happens in this mismatch, this is A and instead of T some G is there, AG base pairing, let us see. So this G is still not methylated on the newly incorporated DNA strand. Methylation takes some time to occur. During that, this mismatch repair system functions. So let us see how it all takes place. If you see now, the methyl mismatch repair system, it involves recognition of the methylation pattern in the DNA bases. Escherichia coli, it methylates its DNA at the palindromic sequences, and these palindromic sequences are GATC. This is the GATC, as you can see there, using an enzyme called as deoxyadenosine methylase or the DAM methylase. So this DAM methylation system is called as the deoxyadenosine methylase. It adds a methyl group to the adenine in the both strands of the sequence. So in both the parental DNA, adenines are methylated wherever there is a GATC palindromic sequence. So these methyl groups are added on adenines. Adenine and thymine normal base pairs are there, and on this adenine there is a methyl group added. Now if you see the DAM methylase, which is called as the main system of the cell for error correction, the methylation is there on the parental strand but it is not present on the newly synthesized strand. This DAM methylation, it takes some time. The methylation system of the prokaryotes, it takes some time to methylate the newly formed DNA, and in that time if it doesn't methylate, the error remains. Now this is consider a and a G mismatch which is there. Now the difference in this methylated and unmethylated strand, the black is the parental strand which is methylated and the purple is the unmethylated newly formed strand. This difference in the methylation, it allows the cell to distinguish the correct sequence, okay, and correct the wrongly incorporated base. How does that thing happen? As you can see, this A which is there, it is not methylated on the base strand. This is the recognition point. Many bases away from the mismatch, that is, there is a mismatch on which one base is correct, A is correct. Correct in what terms? Because it is present on the parental strand, therefore it is correct. And the one which is on the non-methylated strand, that is recognized as incorrect. Many bases away from this, there is a palindrome GATC on which one of the A is not methylated. That becomes the starting point for degradation of the DNA strand. So you can see now this incorrect base and some bases away. So here what is happening is a series of proteins called as mut-S, mut-L, mut-H, they act together and they repair the mismatch. Now out of this mut-S, this first binds to the mismatch area, then it recruits, or you can say it calls mute L and mute H to come over here and bind and remove the strand from this point to this point. Now this cannot be in a linear fashion, so it loops out. You can see in the animation right now, I have paused it, I will play it. You can see it loops, and in a looped fashion helicase comes, it unwinds the DNA and a exonuclease will cut the DNA. So you can see mute S first binds to the mismatch, then it recruits mute L and mute H. Mute L recognizes the methylated strand and it brings out a loop. Okay, now it brings out a loop. Mute H then cleaves, there is a small cleavage. Now the helicase comes, which is a product of UER-D protein. As it goes on cleaving the DNA, behind it exonucleolytic activity is there, it cleaves, and behind it DNA polymerase 1, it simultaneously fills the gap, it goes on correcting, and then in the end ligase will come and it will seal the DNA, whatever the gap is remaining, that will be sealed. So with this the error is completely removed. There is a complete removal of the DNA error, that is the mismatch is removed here. So you can see the proteins which are there. Now before going to the PowerPoint version of explanation of this methyl mismatch repair, I would like to give you a repeat of the animation which I just showed you, and also a second animation where you may get a broader perspective and a better understanding. I hope you enjoy this animation first, and then we will move back to the explanation and our discussion. So I hope you enjoyed the whole process in animation. Now let's see the explanation in detail about methyl mismatch repair. This is one of the types of DNA repair systems in the prokaryotes as well as in eukaryotes. This methyl mismatch repair functions with DAM methylase enzyme, that is the main enzyme which is used for methylation during replication process. Apart from this, mut H, mut L, mut S proteins, as you saw in the animation, DNA helicase, that is the one which unwinds UERD protein, SSB protein, DNA polymerase 3, exonuclease 1, exonuclease 7, REJ nuclease, exonuclease X, DNA ligase. These enzymes across all domains of life forms bought at one place, enzymes, these function for the mismatch repair system to function. So whenever there is a mismatch in eukaryote or prokaryote, all these enzymes, these proteins, they assemble there. These not all at the same time, there will be different enzymes required in different organism, and this differs with different genera and species. This all collectively, if you say the mismatch repair proteins and enzymes, these are the ones which are involved in the repair of the damaged DNA. So basically, if you see what is a mismatch repair, this mismatch we have seen that is a abnormal base pair. Instead of AT there is a GT, let us say, and downstream there are some methylated palindromic sequences GATC, GATC. So GAT and C, so follow the red cursor, GATC. So this GATC 5' to 3' and GATC 5' to 3' on the opposite end. So here these two methylated adenines, these are the recognition point for repairing this. So here it will be cut until this end, there will be removal of the bases. So this is a system. How do you define a mismatch repair? We define this mismatch repair as a system of recognizing and repairing the errors which occur during DNA replication and the repair errors caused by DNA damage. There are many different types of mismatch repair systems, but the mismatch repair mechanisms, they are strand specific, means they don't act on the strands randomly, they are very specific. As I have said here, they start from the palindromic sequences. Now the main challenge for the proteins to repair is to scan the whole DNA and sense the distortion. Okay, that we saw is done by mut S, which is a special type of protein, and the discovery of this mut S was very significant in the history of genetics very recently. So the major challenge for this enzyme system is to scan the genome for mismatches, repair before the next round of replication takes place. Otherwise, once the methylation corrects this mismatch, it will remain there as an error, and in the next rounds of replication, AT will be converted to GC, which we have seen in the base substitution type of mutations. So this has to repair the mutation before the synthesis of new strand takes place. Now mut S, this is an amazing protein. This was discovered by Peggy Say, PhD, and she has numerous papers on the function, model, active sites of these enzymes. You can Google it and find out for your own. Now this protein, this is a, or you can say a bio information generated design of a ribbon model of mut S protein. This has some domains. This is domain 1, 2, 3, 4 and 5. So each color is showing a domain, and this is the DNA. This ball model is the DNA, which is a substrate for this protein, and it fits into it, and as it goes it halts there at the, it has a specific shape where it can sense the distorted DNA and it will stop at the point of distortion. Distorted DNA, they are somewhat kinked, means they are separated apart because hydrogen, they do not bond. G and T, they will not bond, so suddenly there is a kink, and this protein stops there. Once it stops there, it invites the mut L and H to come over. So the animation which you just saw, I will explain it, and these points which are there coming on your screen can be taken as notes. That is the main aim why I have given you the PowerPoint presentation here. This matter you can consider it as notes. So we'll go through it once again. See here, there is a methylated strand, there is an unmethylated strand. The DAM methylase enzyme, deoxyadenosine methyl group adding enzyme, which adds at the adenine. So DAM stands for deoxyadenosine methylase. It acts but not immediately on the lower strand. As you can see, the DNA has a late methylation on the new strand. This difference in the methylation allows the cell to correct the sequence by considering the parental strand as a template strand. That will become, means, you see, after removing the bases there should be a template which will be correct and which will be wrong. Just for 1 second imagine if while correction the parental strand is removed and the new strand is considered as a template, then the mutation will remain as it is. Okay, so that is the point to be understood. The parental strand is considered as a template, and for that, what is the yardstick? What is the point to be considered by the enzyme? Non-methylated DNA. The new strand which is yet to be methylated, that is considered as incorrect, the whole strand, and it is removed. So we have seen this in the animation. Now just see what will happen if the cell is DAM negative. DAM negative means if there is no methylase enzyme. If methylase enzyme was not present in the cell, think about the high rate of mutations which will occur. This is a question, higher order thinking question for you in the bottom. If it is hidden by the strip, I would like to put this question which is there in the box orally. Just listen. What would happen to the mutation rate in a DAM negative bacterial cell, a cell which is not having the methylase enzyme? The answer is there will be high mutation rates in such cells. Okay, so DNA MMR in E. coli requires these many enzymes, that is MUT-S, MUT-L, MUT-H, UVRD protein, single stranded binding protein, and exonuclease. So MUT-S, MUT-L, MUT-H, DNA helicase, which is a product of MUT-U or UVRD. There are 4 exonucleases in E. coli: exonuclease 1, 7, 10, and REC-J. Not all are required at the same time, but depends upon which strand is there. Some may act on 5' to 3', some may act on 3' to 5' exonuclease activity. Some may have a dual function, they are bifunctional in nature. Some are having only single function of removing, not adding, so on and so forth. So this is one which you will study in more detail in your post-graduation studies. So the next enzyme is single stranded binding protein, DNA polymerase, holoenzyme. Holoenzyme means you have seen in the animation only the beta and the beta clamp and the one of the subunit is needed. Not the whole DNA polymerase is needed, only the holoenzyme is needed. Apart from that, DNA ligase, MUT-S, MUT-L, MUT-H, they are the ones which initiate the mismatch methyl repair, and we have seen that they have a very important role in survival of the cell in high rates of mutation. So a series of proteins, these MUT-S, MUT-L, H, they act together. MUT-S, it finds the mismatch, binds it, recognizes the methylated strand and brings it to a loop. MUT-H, as you can, you know, that cleaves the unmethylated strand which contains the GATC palindromic sequence. So you can see this all protein they assemble at the site, and a series of protein MUT-S, MUT-L, MUT-H, they recognize the GATC region here, that is the palindromic sequence. Then there is a looping of the MUT-L, it recognizes the methylated strand and brings it in a loop. You can see it brings it in a loop. That's why it's called as MUT-L. MUT, it stands for mutator genes. All these proteins which act on the genes where mutation is there, these are mutator genes. So MUT-L, maybe it is for the formation of the loop. Okay, MUT-H then cleaves the unmethylated strand. So there is a double stranded loop there, out of which one is methylated, which is held by L, MUT-L, and the MUT-H which is there, which is forming a 3 protein complex 2L and H. It is having access to the unmethylated DNA and it introduces a cut. This cleavage is on the unmethylated strand near the GATC region, that is, this is near the palindromic region. Now Then once this cut is there, this cut invites the UERD helicase. This is the helicase enzyme. It starts unwinding the cleaved strand. Recall the animation. It starts unwinding the cleaved strand, exposing it to a variety of exonucleases. Many exonucleases, they are means in different bacteria, of course. In E. coli there are 4 to 5 exonucleases, depending upon the situation, depending upon the gene, depending upon various conditions. Any one of the exonuclease will be invited here, attracted, and it will cleave. This will result into a gap. The gap is filled, as we have seen, by DNA polymerase 1. Okay, the DNA polymerase 1 helicase, it is unwinding. DNA polymerase is filling the gap. The DNA polymerase continues filling the gap, as well as the helicase unwinds the gap. It cleaves the gap till the mismatch is reached. This mismatch has to be covered, and as it goes past the mismatch, the DNA polymerase continues through the mismatch region and inserts the correct nucleotide there. Okay, so for one base, so many bases have to be covered, because from where to start and where to end. So that is the whole array of enzymes recognizing, repairing, and patching it. In the end, when the DNA polymerase falls off, they are all, when the process is completed, they just fall off the uh, strand, and ligase, it seals the gap. Ligase, it completely seals the gap. So here you can say the role of ligase. After a while, DNA polymerase falls off the DNA, and the gap which is remained here, that is the phosphodiester bond, is introduced by DNA ligase. So this is the main role, or this is how at the molecular level it functions. If you want to summarize this in simple terms, mismatch repair system, they remove the single base pair error which escapes proofreading. Remember, it is a single base pair error which is escaping the proofreading, therefore it is very important. Now if it was an induced type of mutation, then there are many other types of repair also. But here what is happening, this may result into a mutation, a spontaneous mutation, which may sustain for the next generation. So it is very one single base. We have seen there are 9 different types of phenotypic effects which can happen on the protein. So a single base pair error which escapes the proofreading is removed by methyl mismatch repair system. This is well studied in E. coli. Proteins are called as mute proteins because they are acting on the mutator region. What is a mutator gene? A gene which is undergoing mutations frequently is called as a mutator gene. So wherever there is a mismatch, that will be called as a mutator gene, and the protein acting on that mutator gene is called as a mute gene. So mute S recognizes the mismatches, mute H, it nicks one of the strand ahead of the mutation. A helicase and a exonuclease are recruited to remove the base pairs including the mismatch. DNA polymerase 3 will fill the gap, and ligase will seal the gap. This is all in general about the methyl mismatch repair system. I hope you understood it. I hope you enjoyed it. This, though it seems very difficult, repeat the whole process once again and you will understand that it is a very synchronous, systematic, and a very coordinated system, and the recognition is the unmethylated strand. So that's all for now about the repair systems. The mismatch repair system knows there is a mutation on the basis of unmethylated DNA. Okay, you have to remember this thing. Now in the end, I would just like to give you additional information. This is usually asked in the MCQs or the competitive examinations which you will be giving. They will ask you, have you studied mutation and you don't know Ames test? Then that should not happen at the graduation level. So just for information, I would like to tell you, using auxotrophic mutants of Salmonella typhi or Salmonella species, it was discovered by Bruce Ames in early 70s. It is called as the Ames test for detecting carcinogens. What are carcinogens? Carcinogens are cancer causing compounds, and the whole principle of this detection is that all the cancer causing compounds are mutagenic agents. Okay, remember this statement. This is the principle of this Ames test, and Ames test is used to detect mutagens. if there is a mutagenic compound, say you see the cosmetic products, usually all the cosmetic products which are there, I will not name a particular thing, but it is believed that everything which is facial, it may be the mascara, the powder, or the lipstick, or whatever the facials are there, which has a boom in the industry in today's bridal makeup, etc. Today we are having all type of nonsensical makeups called as 3D makeup, and I don't know what is, but all this contain the carcinogens majorly, which are not approved. They contain majorly, I am saying not all, but they contain carcinogenic compounds which are cancer causing compounds, and all carcinogens, many carcinogens, they are mutagenic in nature. They cause mutations in the skin cells, etc. So Ames test is aimed at detecting the carcinogenic compound, whether it is a mutagenic or not, detecting the mutagenic agent in the carcinogenic compound. So it is a simple test where auxotrophic mutants of Salmonella typhi are taken. They are auxotrophic for histidine, means if histidine is not added in the medium, they will not grow. They need histidine. The other is a prototrop, means if you are adding histidine, they will grow. If you are not adding histidine, they will not grow. In the presence of this carcinogenic compound, it will mutate and become auxotroph. So this is a simple test, no need to go in much detail. You remember that Ames test is used for detecting mutagenic agents. How most of the carcinogenic compounds in the market today, it can be anything, it can be food, it can be cosmetics, it can be chemicals, it can be anything which is harmful for the living system of the humans. So those which we consume, means apply to our body, ingest inside our body, eat, medicines, food, if you want to see whether that is mutagenic or not, follow the Ames test. If it is carcinogenic or not, because all carcinogens, cancer causing compounds, are mutagenic in nature. So Salmonella typhimurium, Ames test, and mutagens. In the post graduation studies, we can have a elaborate discussion on the Ames test as a potent process for, or a very powerful process for detecting the mutagenic agents. So that's all for this unit of discussion on DNA mutations and repair. The next series of lectures for the next unit will be the regulation of gene expression. This is a very beautiful concept, a very significant concept. We know that a gene is responsible for production of proteins or enzymes. Now the question is about the quantity and when to produce and when to stop. That is called as regulation. How does the cell know which enzyme is to produce to when and in how much quantity? That is the point of discussion in regulation of gene expression. That will be in the next series. Till then, keep studying, keep aiming for higher domain of knowledge, always have a quest to understand what is next, not forgetting the past, having a very good fundamental base of the understanding what you are studying, and going forward and higher.